microphone play.
All right. Hi, everyone. Maybe we'll get started with the introduction while we wait a couple of minutes for people to come in. So I'm Alice Soragni. I'm an assistant professor in the Department of Orthopedic Surgery here at UCLA, and I call it the CTSI Distinguished Speaker Regenerative Medicine theme. It is my distinct pleasure to introduce today's speaker, which is Dr. Janai Carr-Ascher. Dr. Carr-Ascher received her bachelor degree in biology from UCSD and her MD PhD from the University of Illinois. After her residency in internal medicine at UCSF, she actually pursued a Hem-Onc fellowship here at UCLA. While at UCLA, she joined the laboratory of Dr. Owen Whitty, where she focused on a couple of different things. She pursued a project looking at the role of wild-type kinase dysregulation driving prostate cancer metastasis. And she had a second project, which was centered on elucidating the critical molecular pathways driving sarcomagenesis using a mesenchymal stem cell transformation model. She has continued this work as she started her independent lab at UC Davis in 2019, I believe. Now, this work is actually very important. Sarcomas is an incredibly diverse family of tumors. There are over 100 subtypes. And one of the absolute critical outstanding questions in the field is, what is the cell of origin of these tumors and how a probably shared precursor can give rise to these many and these diverse tumor types. This has been a really elusive question, and Dr. Carr-Ascher and the research has really put us as close as possible to deciphering this elusive cell of origin. It's a groundbreaking work, and I believe she's going to discuss some of this outstanding progress later in her talk. In addition to running her laboratory, she's a physician scientist that sees sarcoma patients as part of her practice. She has really made significant contribution in the field, underscored by the leadership roles she has, as well as her honors. She was chief fellow of the UCLA STAR program when she was here during her training. She is the recipient of the Christine and Helen Landgraf Memorial Research Award. She is a medical oncology advisor for the National Leiomyosarcoma Foundation. And she's also part of the NCCN Guideline Committee, who sets treatment guideline for soft tissue sarcoma and GIST. She currently is the research director for the Hem-Onc Fellowship Program at UC Davis, as well as the assistant director of education, training, and career development at the Cancer Center. She truly, as with her accomplishment, an unprecedented result particularly in the sarcoma field and development of preclinical in vivo models to study sarcoma cell of origin, as well as sarcoma metastasis. She is breaking new ground on finding pathways and drivers that could lead to groundbreaking treatment for sarcoma. And so it's my absolute pleasure to introduce her today. Please.
Well, thanks so much for the kind introduction, and I'm really excited to be here. UCLA has been a very transformative place for me. So I came here after undergraduate and worked in a lab with Joanna Jin in the neurosciences for 3 years when I finished undergrad and before I went to med school. And that was really when I sort of fell in love with basic science. And so my time at UCLA has been really important to me. And of course, I also did my fellowship here and started the research that I'll show you today, working in the lab of Dr. Whitty. So today I'll tell you a couple of stories. One will be sort of the focus, which, as Dr. Soragni mentioned, is really looking at the development of in vivo models to study sarcoma. And then I will also tell you a brief story about an epigenetic regulator that we're very interested in. And so both of these stories have a translational component to them. So I'm excited about the potential in the future for the treatment of patients. So I have no disclosures to make. So for those of you that aren't familiar with sarcomas, this is an umbrella term. So any tumor that arises in connective tissue, whether that's bone, muscle, fat, skin, blood vessels, they fall under this umbrella of sarcoma. So you can imagine that this is a highly heterogeneous set of diseases. And as she mentioned, there's more than 100 subtypes. And so there's this huge heterogeneity across the disease and then also within the disease. So most commonly, sarcomas appear in the limbs. So in the thigh and in the arms are the most common places to find sarcoma, but they really can arise anywhere in the body. So they can appear in the head and neck. They can appear in the retroperitoneum, so the area near the kidneys. And one thing that's really interesting to think about with sarcomas is, as I mentioned, there's these 100 different subtypes. But for example, leiomyosarcoma, which is a smooth muscle type of sarcoma, can appear in the limbs. It can also appear in the retroperitoneum. It can also, appear in the head and neck. And, one question is, are those the same tumor, right? Or are they different tumors? It's also common to find it in the uterus. Is that the same disease? So there's a lot of questions in sarcoma, given the heterogeneity of the disease, both in the histologies and in the anatomical distributions. So when we think about sarcomas, it's actually kind of difficult to think about 100 different tumors altogether. And so in this Nature review, they basically tried to group sarcomas by their histologies. And so let's see if I can use the pointer here. So as you can see over here, they put together all the blood vessel tumors. And then over here, they put together all of, sorry, I can't see well. Over here is all the skeletal muscle tumors, et cetera. So they tried to group them by histology. And that's one way to think about them. But the reality is that that's clinically not very relevant. So we don't treat angiosarcoma, right? That's right here. We don't treat that the same as the tumor right next to it, epithelioid hemangioendothelioma. These are very different tumors with very different treatments. So I think about sarcomas as being either bone or soft tissue sarcomas. And this is clinically important because bone sarcomas, we do actually have different treatment regimens for those. And those are more common in children than they are in adults. And then within these soft tissue sarcomas, we think of them as either being low grade or high grade. And so low-grade sarcomas really are a surgical disease. So they're resected by the surgeon, and to be honest, those patients never make it to me, which is good for them. So that's really a surgical disease. But what I'm interested in are these high-grade sarcomas, the high-grade soft tissue sarcomas. And within this group, they're genetically divided into two groups. So on the one side, you have the sarcomas that are driven by a translocation, and these we refer to as having simple karyotypes. And so the most common example of that is Ewing sarcoma that's driven by the EWSR-FLY1 translocation. And then there are complex karyotype sarcomas that really are characterized by having a complex karyotype. So they have a lot of aneuploidy, a lot of chromosomal gains and chromosomal losses. And because these tumors, these complex karyotype tumors, are the tumors that are most common in adults. More than 80% of adults will have complex karyotype sarcomas. That really is the focus of my research as I'm an adult oncologist. So when we think about these tumors, there are actually more than 70 subtypes of high-grade complex karyotype sarcomas, but these are the most common, and so the ones that we'll talk about today. And you can see that these histologically all look pretty different. So the most common subtype in adults is undifferentiated pleomorphic sarcoma, which basically means it doesn't stain for anything. We can't tell what this tissue of origin is. It doesn't stain for any of the muscles, the muscle markers or bone markers, et cetera. And that's actually the majority of tumors in adults. And then there's dedifferentiated liposarcoma, leiomyosarcoma, which is a smooth muscle tumor, and then myxofibrosarcoma that is probably somewhat related to the undifferentiated pleomorphic sarcomas. But you can look at these tumors histologically and see how different they are, really. And one thing that is clinically very challenging is that when I see patients with these different subtypes, I essentially treat them the same. And that's primarily a numbers problem. So about 13,000 people this year will be diagnosed with sarcoma. About 5,000 people will die this year of sarcoma. Whereas in breast cancer, about 270,000 people will be diagnosed this year. So when you're trying to run a clinical trial and when you're trying to investigate drugs, there are significant challenges in sarcoma, right? Because you only have 13,000 patients. The majority are not treated at academic centers that have clinical trials. And you can imagine if you have 70 different subtypes and you take 13,000, start dividing it by 70, you get into some pretty small numbers. And so what we've done historically is basically run trials by grouping together all of the sarcoma patients just out of necessity. to have enough power to run the studies. And then in the end, the studies are somewhat equivocal. And then we try to do subtype analyses to figure out which types of treatments to use for which subtypes, but the studies aren't powered to look at that. So in the end, we've really continued to use the same chemotherapy regimen for the last, more than 30 years for these types of sarcoma. And so it's clinically very challenging and something that my lab is focused on trying to better understand what the relationships are between these sarcoma subtypes. How are they similar? How are they different? Which groups should we put together for trials and which groups should we not put together for trials? Because the reality is, when you look at these histologically, they may be as different as lung cancer and prostate cancer, right? We just really don't know. And so that's really what I'm focused on better understanding. And so, as I mentioned, the clinical problem really is that sarcomas are rare. And so this limits our clinical trial options. Our patients receive uniform treatment for heterogeneous disease. And about 35% of patients who present with a localized tumor, so in a curable situation, about 35 of those patients within the next 5 years will develop metastatic disease. So our treatments are just not that effective as seen by this recurrence rate. So again, I'm really focused on really trying to understand how to put these together and how to take them apart. And so I started by trying to determine whether or not we could create a model system that will allow us to study these different sarcoma subtypes. And essentially, if we could start in the same place, start with the same cell and develop different types of sarcoma, then we could better understand the relationships potentially between them. So that was really the question, is whether or not we can develop models that reflect this high-grade complex karyotype and recreate this disease spectrum. And so I started this work when I was in Owen's lab here, and there was a postdoc at the time, Jung-Wook Park, who was using a forward transformation model to develop small cell prostate cancer. And so I know the slide's a little busy, but basically what he was doing was taking normal human prostate cells. So these are not malignant cells. And so he was separating out the basal cells here, separating out the basal cells and then adding a series of genes. So basically adding MYC, BCL2, an activated version of AKT, and then knocking down RB and P53. And when he added in this 5-gene combination that he called PARC-B, you essentially can develop small cell prostate cancer starting with these basal cells. And you can see that by using different combinations of these genes, you can also develop an adenocarcinoma phenotype as well. And so I started out by seeing if we could do the same thing in sarcoma. So can we take these gene combinations and essentially try to create sarcoma from mesenchymal stem cells? And so mesenchymal stem cells are really the accepted cell of origin for sarcoma, although there's a lot of debate out there as to whether or not all 70 to 100 subtypes arise from the mesenchymal stem cell. It is unlikely that that's the case. And there are likely, progenitor cells downstream that give rise to certain subtypes. But I think in general, it is the accepted cell of origin. And that's because it does give rise normally to bone, muscle, fat, et cetera, in the body. So it is thought that these are the cell of origin. So I tried to take Jung-Wook's approach and taking mesenchymal stem cells, of course, instead of prostate cells, and seeing if we could make genetic manipulations in order to develop different sarcoma subtypes. So I'm not the first person who has attempted this. So this is a summary from a review article where people have developed some transformation models. And what you can see is that people have taken mouse mesenchymal stem cells and have been able to make histologically different subtypes of sarcoma. And they can do this just by inactivating tumor suppressor genes. And then these are transgenic mouse models. So essentially the P53 knockout mouse or the P10 knockout mouse, they do develop sarcomas. They also develop other types of cancer, but they will develop sarcomas. But what's been really challenging is actually using human mesenchymal stem cells in order to drive transformation. And so it's been tried to just knock out RB or knock out P53, and human mesenchymal stem cells do not transform into sarcoma just with the loss of P53 or RB. You really have to add a strong driver. And so in the past, people have added viral oncogenes And this has been effective, but the tumors that are created don't actually reflect the human disease, right? Because human sarcomas are not virally driven tumors. And so I wanted to sort of build upon this work. But one thing that I'll mention here is that one major limitation, it's thought that why it's very difficult to transform human mesenchymal stem cells is because of differences in telomere maintenance. And so it's thought that you really have to disrupt or activate TERT in order to transform human mesenchymal stem cells. And so with that in mind, I went to the TCGA data. So I'm sure everybody's familiar with the Cancer Genome Atlas, but this is the a figure from the sarcoma TCGA data, where essentially they looked at 6 different subtypes of, and you can see them down here, but they looked at 6 different subtypes of complex karyotype sarcomas. And then they looked at synovial sarcoma, which is a translocation-driven sarcoma that's the most common in adults. And so if you look, I know this figure is a bit busy, but if you look at the histologies across the top, you can see that they're looking at myxofibrosarcoma, undifferentiated pleomorphic, those that I mentioned to you. And they looked at whether or not there were genetic mutations that were commonly seen across the sarcomas. And you can see that a lot of the tumors do have mutations in P53, ATRX, and RB, but that's not all of the sarcomas. So interestingly, sarcomas actually have a lot of aneuploidy, but they actually have a fairly low rate of mutations. And the aneuploidy is reflected here. So essentially all of the red are chromosomal gains, all of the blue are chromosomal losses. And so you can see that there's quite a bit of aneuploidy here, and it seems to be fairly recurrent. So there are some genes here that we commonly see being amplified. And then you can see that P53 and RB, ATRX can be lost at the chromosomal level, even if they aren't genetically mutated. So taking all of this information, taking the idea that you have to disrupt telomere maintenance, that we don't have many genetic mutations, but we do have amplifications and losses of certain tumor suppressors. We took that information and tried to apply this to mesenchymal stem cells to see if we could develop sarcomas. And so this was the model. So ASC 52 telo is a immortalized mesenchymal stem cell line. So it is immortalized by TERT. So it does have that, the telomere, it's sort of taken the telomere issue out of the equation. And so this is an immortalized mesenchymal stem cell line that was derived from adipocytes. And then we used CRISPR-Cas9 to knock out RB and P53. And so the advantage to using an immortalized line is, of course, it can grow indefinitely in culture. So we did attempt this with normal mesenchymal stem cells, and there are a lot of technical issues that are have been difficult to address, but we are circling back to work on some of those. But for now, we have used CRISPR-Cas9 to knock out RB and P53. And one thing that's interesting is P53 is lost. It's a heterogeneous loss. Yes, it's a heterozygous loss. And that's because the complete loss of P53, for whatever reason, is not tolerated by these cells. So those cells tend to undergo apoptosis for sort of unclear reasons. But we've taken the RBP53 cell line, and then we added a lentiviral library to these cells. And essentially, these were factors that we saw in the TCGA data that were overexpressed or things that are known to be amplified in sarcoma. And so we added those in as a lentiviral library. And what we saw was that we were able to develop histologically high-grade complex karyotype sarcomas. So when you look under the microscope, what we see on low power is you can actually see sort of 2 different histologies here side by side. And when you look at them on higher power, you can see that this is just sheets of cells. And then this seems to have some sort of stromal component to it. And so we were obviously excited to see that we were able to develop sarcomas, and these histologically were confirmed by our pathologist that these are high-grade sarcomas. We wanted to know if phenotypically these match the human disease. So what is probably the most interesting thing about sarcoma is that there are, these 70 to 100 different subtypes, and they only really have one thing in common. and that's that they metastasize to the lung. They do not, despite the fact they develop in the thigh, they do not metastasize to the liver. They seem to bypass that system, and they really metastasize almost exclusively to the lung initially. And so we implanted these cells into the calf of a mouse instead. So initially we made these subcutaneously, but then we injected cells into the calf of the mice. And what you saw was that within a relatively short period of time, just in a few months, we were able to develop lung lesions. And so these mice did not have tumors in their liver or elsewhere in the body. So it does seem like they are phenotypically reflecting the human disease as well. So then this was a screen, of course, so we wanted to figure out which genes were actually driving the sarcoma development. And so we did barcode sequencing, and there were 21 factors in the library, and we did barcode sequencing, and what you can see is that 3 factors, so DDIT3, KRAS, and YAP, were significantly enriched in the tumor cells. And you can see the starting material across the top here. So you can see that essentially, the YAP is actually pretty low expression in the starting material and then significantly, amplified in the tumors that form. So then we wanted to take these factors individually and see if we could create sarcomas with these individually. And so what you can see here on the left is that if you add YAP alone to our RBP53 cells, you develop undifferentiated pleomorphic sarcoma. And this is evidenced by the lack of staining of smooth muscle actin and myogenin. So this is a muscle marker and this is a smooth muscle marker. And it does light up blood vessels, but we don't see any expression elsewhere. And then if you add KRAS into the system instead of YAP, you get this myxoid phenotype. So this essentially creates myxofibrosarcoma, whereas YAP is creating undifferentiated pleomorphic sarcoma. And then we went back to our primary screen because YAP and KRAS are such strong drivers of proliferation that we suspected that there are probably other drivers within the population, but they were being masked by the high proliferation rate of YAP and KRAS. And what I will also mention is that DDIT3 is really interesting because it consistently comes up in our barcode sequencing, but on its own, it is not able to give rise to tumors, even over a 1-year period. So while it keeps appearing, it does not seem to be independently a driver and likely helps to facilitate sarcoma development by other factors. So we went back to the screen and repeated it and removed YAP, DDIT3, and KRAS. And then we were able to identify a series of other genes that were significantly enriched. And so we put those individually into our model. And I won't go through all the details here, but what was interesting is that CDK4 gives rise to a pleomorphic leiomyosarcoma phenotype, as you can see by the positive staining of smooth muscle actin. And then osteosarcoma can be driven by PI3 kinase. And so this was really interesting because these are actually subcutaneous tumors, and you can find the development of bone here. So this bright pink material is bone, and you can see the tumor cells embedded there. And it stains positive for SAPB2, which is a fairly specific marker for osteosarcoma. So this is just to summarize all of the data that we generated from the screens. So essentially, you can see all the genes listed here in the latency period. So obviously, some of these are really robust drivers of sarcoma development, such as KRAS, where you can form tumors just in a couple of months. And then we also calculated the efficiency. So essentially, of all the cells that were implanted, how many actually gave rise to tumors? And so some of these are really robust drivers, right? Like June is actually an excellent driver of sarcoma development, whereas others, like cyclin D, is really a pretty poor driver of sarcoma. And this may actually be spontaneous transformation. So we were able to identify robust drivers of sarcoma development and to identify different histologies. So essentially, KRAS was able to give rise to myxofibrosarcoma, YAP to UPS. And then I will say that the CDK4 and PI3 kinase, while they do give rise to leiomyosarcoma and osteosarcoma, it's not 100% of the time. Sometimes we do get undifferentiated pleomorphic sarcoma instead. And we're working on trying to understand why that is, if it has to do with the starting material or potentially other factors. Well, then we wanted to look and see whether these models also genetically reflect the human disease. And so all of this work is done in collaboration with Tom Graber here at UCLA and his graduate student, Jack Freeland. And so they looked at, we did whole exome sequencing of the tumors and looked to see whether or not the tumors have this aneuploidy phenotype. And so this is a heat map here on this side, looking at chromosomal gains and losses. But I'll direct you over here to this copy number alteration score. We essentially set the parameters by looking at thyroid carcinoma, which has very low rates of aneuploidy, and then kidney cancer, which has very high aneuploidy scores. And then we plotted the human TCGA data here. So you can see that within the human disease, there's quite a heterogeneous amount of aneuploidy. And then we plotted our models as well as the starting material. And so as you can see, the starting material has a fair amount of aneuploidy at the beginning, which is probably part of the reason that this system is amenable to transformation. But you can see that there's quite a bit more aneuploidy that's noted in our model systems driven by KRAS, CDK4, and PI3 kinase. And then one really interesting observation has to do with YAP amplification. So what we saw in the whole exome sequencing is that if you look down here at the starting material, so this is looking only at chromosome 11, and then we've put a line here in yellow as to where the location of YAP is on the chromosome. And you can see that in the starting material here, there's not amplification of YAP. So we don't see that there, nor in the KRAS model. So in KRAS-driven tumors, we do not see any amplification of YAP or that area of chromosome 11. And if you look at the YAP model as expected, you would see what appears to be amplification of YAP, which of course is the exogenous expression. But what's interesting is that the CDK4 and PI3 kinase tumors have actually amplified the portion of the chromosome that contains YAP. And so if you look over here at the absolute amount of mRNA expression, you can see that the amount of YAP in these models is extremely high, and that the copy number is up to 40 or 50-fold. So we do think that YAP is extremely important in the development of some subtypes of sarcoma and may create an interesting treatment opportunity as there are inhibitors of YAP that are in development and some in trials. So the lab is working on better understanding the mechanisms of YAP-driven sarcoma. So more to come on that in the future. And just for those of you that are not as familiar with YAP, so YAP is the downstream transcription factor in Hippo signaling. So when Hippo pathway is off, YAP is not phosphorylated and is able to translocate into the nucleus and bind to its preferred binding partner, the TEAD family, and drive gene expression. And then when Hippo signaling is on, YAP is phosphorylated and degraded. And so what's been observed in sarcomas is that patients with high nuclear levels of YAP and its paralog TAZ have poor prognosis and higher metastatic rates. So we're very interested in further exploring mechanisms of YAP-driven sarcoma development and progression. So then we wanted to look at our models a little bit closer. And so this is RNA sequencing data. And so this is the TCGA data where essentially we've taken different human tumor types and projected them into a PC space and then projected our models onto that. So you can see that this purple circle here is the sarcoma TCGA data. And then these are our models here in this gold color. And so you can see the overlap at the RNA level is not perfect, but it is fairly close to the human sarcomas. And then we looked more closely at YAP and KRAS-driven models specifically because these models histologically were so reliable and they're very robust. And so we really focused in on YAP and KRAS because of their consistency. And so if you look at YAP, and so this is a rank plot where basically you take genes that are highly expressed in the model, and then you take genes that are highly expressed in human UPS, and then you overlap them and see if you if the gene expression patterns are able to be overlapped. And so if you look here at this dark orange and then at this dark orange, you can see that YAP overlaps nicely with the undifferentiated pleomorphic sarcoma human samples. And then KRAS overlaps nicely with the human myxofibrosarcomas. So it does seem as though the gene expression patterns are similar between our models and the human disease. So then we looked a little bit more closely at our YAP and KRAS-driven models and looked at what pathways are upregulated. And so this is gene set enrichment analysis looking at UPS or looking at YAP and UPS, and then KRAS and myxofibrosarcoma. And so this data is interesting because it's actually an overlap of our model with the human TCGA. So we basically focused in on things that were upregulated in both. So what you found was that oxidative phosphorylation pathways are highly enriched in our model and in human undifferentiated pleomorphic sarcoma. And then you see these DNA damage pathways are highly enriched in myxofibrosarcoma and in our KRAS model. And so the oxidative phosphorylation is really interesting because there is a compound actually that went through clinical trials that is an oxidative phosphorylation inhibitor. And when I first thought about an oxidative phosphorylation inhibitor, I just assumed it would be too toxic to be used in patients. But realistically, the side effects were actually pretty minimal. The drug wasn't terribly effective, but the side effects were pretty minimal. So I'm just showing you here an example of one cell line, but so now we're moving out of our model and moving into sarcoma cell lines. And we treated them with an inhibitor of Hippo signaling. So verteporfin is known to disrupt the interaction between YAP and TEADS. And so it's a very well characterized Hippo inhibitor. And so when we treat our cells with either a Hippo inhibitor or an inhibitor of oxidative phosphorylation, we actually are we can pretty robustly inhibit proliferation at fairly low drug concentrations. And so we were interested in whether or not these cells that tumors that overexpress YAP may be more sensitive to oxidative phosphorylation inhibitors. And so perhaps the combination of the two would be a potential clinical approach. And so this again is just looking at a panel of cell lines. So these three cell lines are all undifferentiated pleomorphic sarcoma. And then this is a leiomyosarcoma and a dedifferentiated liposarcoma. And so you can see we've treated the cells with either the oxidative phosphorylation inhibitor, the Hippo inhibitor, or the combination. And you can see it's not effective for every tumor type. For example, UPS2, we don't see any synergy here, but here in the UPS lines and especially in the dedifferentiated liposarcomas, we are able to see a nice synergy with the combination of Hippo and oxidative phosphorylation inhibitors. And so this is something that we're further investigating and hope to be able to translate this in the near future. So now I'm going to switch gears just briefly to tell you, sorry, how much time is that? I just want to make sure I'm not, okay. Great. So now I'll just switch gears briefly and tell you one other story about an epigenetic regulator. And so when it comes to sarcoma, as I said, it really is a surgical disease. And so the greatest driver of patient mortality is actually the surgery. So patients who are able to have a surgery where you can remove a wide margin of tissue around it, that is the primary driver of their mortality. Patients who can have their tumor removed entirely have a much better prognosis than those that cannot. And when you can only remove a part of the tumor, you actually can't make up the difference by giving patients chemotherapy or radiation. They still have lower survival than if they had a surgery with good resection. So the standard of care really is a wide margin surgical resection. And then a lot of patients will have radiation, and then a subset will have chemotherapy. But despite this really aggressive treatment approach, and for the patients, this is about a little over half of a year of treatment, right, of them either undergoing radiation or chemotherapy. It's a very intensive treatment course. And then, despite all of this, they'll still have about a 30% chance of dying of their disease within 5 years. And so this mortality really is driven by treatment failures and the development of metastatic disease. And so we are interested in the cancer stem cells and in sarcoma stem cells because these patients will often have a year or 2 years, where the disease is undetectable, but then obviously it will recur with time. And so likely this is due to a stem cell population that is able to resist treatment and sits dormant for some period of time and then begins to grow. So the cancer stem cell theory, I'm sure all of you are familiar with it, is that within every tumor, there are cells that have a more stem-like phenotype and that are able to essentially give rise to a second tumor with time. And so we were interested in looking specifically at the cancer stem cell population and specifically in different subtypes of sarcoma. And so we developed some resistant cell lines. So most patients who are treated with chemotherapy get an anthracycline. So doxorubicin is commonly given as the chemotherapy of choice. And patients who have resistance to doxorubicin have terrible outcomes and tend to develop metastatic disease very quickly. So a postdoc in my lab, Ed O'Donnell, created a series of anthracycline resistant cell lines by culturing them for long periods of time. And what you can see is that this UPS cell line, whether it's doxorubicin resistant or wild type, the histology is unchanged. So the tumors really look identical under the microscope. And as you can see, the cells are resistant to doxorubicin. And what he looked at specifically was the ability of the cells, these doxorubicin resistant cells, to form colonies in soft agar, but also looked at the stem cell population. And we did this using the aldofluor assay. So this is the idea that that stem cells have a higher levels of ALDH, and essentially you can use this as a marker to identify a stem-like population. And this has been used across a series of tumor types, not just in sarcoma. And so we decided we wanted to sequence a series of sarcoma stem cells because in sarcoma, we really have not been able to identify markers that can consistently identify cancer stem cell populations. We have not been able to sort of look across a spectrum of sarcomas and make comparisons between cancer stem cell populations. And so we were interested in doing this and taking these most common sarcoma subtypes, isolating the stem cells, and then doing a genetic characterization. And so as you can see in this figure, there are a lot of markers that are known in certain sarcomas to mark a stem cell population. But when you try to move that marker to a different sarcoma subtype, so for example, CD133 is a good marker for undifferentiated pleomorphic sarcoma stem cells, but it does not identify leiomyosarcoma stem cells or others. So we really focused on just using aldofluor as this seems to be the only uniform marker across sarcomas. But of course, this likely has its limitations as well. But we wanted to be able to sort of compare a similar population across a series of sarcomas. And so we focused in on these 5 subtypes, so leiomyosarcoma, myxofibrosarcoma, rhabdomyosarcoma, undifferentiated pleomorphic, and liposarcoma. And so we sorted out aldofluor dim and aldofluor bright populations, and you can see that the bright population does form colonies in soft agar. And then we put them through RNA sequencing as well as whole exome sequencing and ATAC-seq. And I won't get into the details of the genome of the RNA sequencing too much, but what was interesting is that between the bright and the dim population, there really isn't a significant difference. There are very few genes that are actually different between the bright and the dim population within each subtype. But one pathway that we saw that was enriched was actually the EZH2 pathway. So in bright cells, so what we think are our sarcoma stem cell population, we do see increased enrichment of EZH2. And what you can see over here is that patients that have, this is plotting patient data from TCGA, is that patients that have low levels of EZH2 have better survival as compared to those with high expression of EZH2. So EZH2 is part of the PRC2 complex, so the polycomb repressor complex. And so EZH2 works with SUS12 and EED to basically add methyl groups to histone 3 K27. And this basically represses transcription and leads to compaction of the chromatin in the area. So this is an epigenetic regulator, and it has been shown that EZH2 is important in other stem cell populations as well, not just in sarcomas. So this is RNA sequencing comparing this is actually ATAC sequencing, comparing the aldofluor bright to dim population. And so this is called a Circos plot. And so I know it's, so basically you plot all of the chromosomes, and then you identify areas that are open. So ATAC sequencing is a way of marking open areas of chromatin. And so it basically integrates a transposase there, and then you can sequence that area and see what areas of the chromosome are open. And so obviously this is a very small and hard to sort of interpret, but what's important about this is that if you look across these different sarcoma subtypes, it seems as though their ATAC sequencing profiles are actually very similar. So it seems that across the stem cell population, the same areas of chromatin appear to be open and closed. And then if you look over here, so this is looking at, again, areas of open and closed chromatin. And so these areas actually, I know it's a little bit hard to interpret, but the blue is actually the areas that are open, and the red are the areas that are closed. And so essentially, if you look at the ATAC-seq profile, and then you compare it to the H3K27 ChIP-seq, you can see a significant overlap between the areas of chromatin that are closed and the H3K27 ChIP-seq profile. So essentially what we're seeing here is that it seems as though EZH2 is likely modifying the chromatin similarly across these different sarcoma stem cell populations, and doing this through H3K27 methylation. And so there is a EZH2 inhibitor that's been approved for a specific type of sarcoma, but that's epithelioid sarcoma that's known to have I&I loss. So basically, it's a downstream regulator. So that is downstream of EZH2. And so it's only approved for that type of sarcoma and hasn't really been shown to be effective across a variety of other tumor types. But tazemetostat, so this inhibitor of EZH2, which is very well tolerated clinically, we decided to treat our cells with this inhibitor of EZH2 to see if this actually does modify the stem cell population. And so what you can see down here is that if you just take our cells and then you treat them with low doses of tazemetostat, you can see that essentially you can decrease the stem cell population. So by about threefold. So it goes from about 10% of the population to about 3% with the treatment of tazemetostat. And then if you remove the tazemetostat from the media, you see a relatively rapid re-expansion of the cancer stem cell population. And then again, if we treat the cells with higher doses of tazemetostat, you see an even further reduction in the amount of stem cells that you see within the cell line. And then again, if you remove it, you can increase the stem cell population. So it does seem as though tazemetostat may be a specific inhibitor of the cancer stem cell population in sarcoma. And so we went back to the doxorubicin resistant lines to see if treatment with tazemetostat would actually resensitize the cells to doxorubicin. And what you can see if you focus on the right here is that treatment with doxorubicin in our resistant lines, as expected, really doesn't change the cell viability. But then if you treat them in tazemetostat, you also do not see any changes in viability. But the combination of the two does seem to be able to resensitize these cells to treatment with doxorubicin. So essentially, in conclusion, we have worked to develop a forward transformation model of these complex karyotype sarcomas. And we think that this model does histologically, phenotypically, and genetically reflect the human disease, and specifically the undifferentiated pleomorphic sarcoma and myxofibrosarcoma. And we're working to sort of refine the other phenotypes and to see if we can expand on this to create additional subtypes of sarcoma. And so as an oncologist, I think that what's clear to me, in this work and just clinically, is that the heterogeneity of sarcoma is a real clinical challenge, and that really the only way I see forward is for us to be able to identify sarcomas within each subtype that are going to be sensitive to specific treatments. So I think we have to move away from thinking about leiomyosarcoma and treating all leiomyosarcomas the same. And instead, we need to do genetic profiling or determine assays that can identify specific sensitivities that different sarcomas will have. So, I would like to develop a YAP signature and to be able to identify tumors that are reliant on YAP and potentially would rely on oxidative phosphorylation. And then we can try to use these combination therapies for those patients that seem to have, the right profile. And also looking at EZH2, it is also possible to develop an EZH2 signature and to be able to use potentially, combinations of EZH2 inhibition with other factors or with chemotherapy in order to try to treat resistant disease and to try to more effectively treat our patients up front. So I really appreciate all of your time, and I look forward to a discussion with everybody. I just want to make some quick acknowledgements. This is the group of mentors that I have at UC Davis who have been very supportive, especially during COVID and other times. They've been a really great group of mentors. And then these are the members of my lab, past and present. And we've worked quite a bit with the Genomics Core facility at Davis to do a lot of the sequencing related to the EZH2 data. And then here at UCLA, I work closely with Tom Graber and Jack. And then, of course, Dr. Whitty was my mentor while I was here before and was nice enough to let me study sarcoma in his prostate cancer focus lab. And then Joanna Jin, as I mentioned, I worked with her previously at UCLA, and she's just been a great mentor over time. And then clinically, I've spent a lot of time with Arun Singh and Bartosz Chmielowski, and they've been my clinical sarcoma mentors, and I still rely on them intermittently to help me out with difficult cases. So thank you, everyone, and I'm happy to take any questions.
Regarding metastasis of sarcoma to the liver, you said they don't go to the liver. Is the mechanism due to lymphatic, does the liver contain lymphatic circulation? And could it be used for this mechanism to control metastasis to other organs?
Yeah, that's a great question. And we actually don't really know. It does not seem as though sarcomas travel through the lymphatic system very much. So the majority of sarcomas, even when they have large tumors in their thigh, the lymph nodes will be clean. There won't be tumor cells growing in the lymph nodes. And so likely it's a hematologic spread of the tumors, but we don't. So it's thought that some cells make it to the liver, they just don't survive there. And so it's not really known why the liver is not a hospitable environment for sarcomas. So, yeah.
Regarding other tumors.
Other tumors.
Sarcoma or some other tumors?
Oh, I'm sorry. So this is specific to sarcomas. Yeah, this is really a sarcoma phenomenon, which is really interesting. Yeah. I think that.
But in the
Yes,
meantime, maybe you can ask a
yeah.
question. So it was surprising me for our wedding, histologically and protein marker-wise, your models look just like the humans, but RNA-seq-wise, they don't really mention it as well as we would expect. Do you have an explanation for that? Or?
Yeah, that's a great question. And we don't know exactly why they don't match quite as well. I think some of it again is the heterogeneity. So I think, we're really making the same type of UPS, right? But realistically, UPS is a disease spectrum, right? And myxofibrosarcoma is a disease spectrum. But I think because we have single drivers, we're really just sort of creating the same type, and it may not, completely overlap. So I am working on a whole genome screen to see if we can try to identify other drivers and then see if some of those sort of genetically better reflect the human disease. I think the CDK4 and PI3 kinase don't really reflect as well because it's a mixture. The tumors, while the majority of the tumor looks like osteosarcoma, there are some areas that don't, that look more like UPS. So I think that's part of the reason too, we're not seeing as good of overlap with those. So I think if we can figure out how to sort of clean up those models, we might see some better profiles.
So we have a question online from Aaron. It's saying, great talk. How often is the Hippo pathway altered in sarcomas? And is this more of an issue in specific subtypes?
Sorry, can you read the beginning again? I'm sorry.
So the Hippo pathway, how often is this actually altered in sarcoma? Like, is it is known? And is this more of an issue in specific subtypes?
Yeah, so there's only been a couple of papers that have looked at YAP and TAS in human sarcomas, and they looked across a spectrum of tumors. So they did not identify any specific subtypes that had higher correlation with YAP. It is known that there's a translocation-driven sarcoma that has TAS as part of the translocation. And so there has been some interest in using Hippo inhibitors in that tumor type. So that's this EHE, this epithelioid hemangioendothelioma, but that's sort of separate. So I guess to answer his question, it seems as though there are subsets within each sarcoma subtype that have high nuclear YAP, but it's not really known, it's not sort of a pan histology finding.
Thank you. Yep. Great talk.
So the inhibition of Hippo pathway and oxidative phosphorylation, have you tested that in preclinical models? What are those well tolerated combined together? And do you see inhibition in metastasis to the lung? I think you mentioned that's the most common site.
Yeah. So that is definitely something we're working on. So the mice are implanted and we'll try. So it seems as though the combination therapy seems to be tolerated in vivo by the animals, but we'll see if it's effective and if the doses are going to end up being high enough. It is a little bit difficult to get high enough doses. So more to come on that, but we're working a lot on the metastasis and trying to expand out that project. And we have some interesting RNA-seq data, and so hopefully I'll tell you more about that, maybe at my next visit. But the metastasis is sort of a work in progress, but we have not done any therapeutic testing using the metastasis model. We're still working on kind of developing that model a bit more.
Maybe I'll go with one more question, sorry, just real quick. So was MDM2 amplification part of your drivers? Does it not come up? Because that would be something that I guess you would expect to be a strong driver.
Yeah. I actually expected to see some liposarcomas because we start with an adipogenic line, but we really see no fat. And so I have not added MDM2 alone. It's something that we're interested in doing, but I haven't done that. It was not part of the screen.
It was not part of the screen. Okay.
No. Yeah, we ended up not including it.
All right. Any other question? Well, if not, let's thank our speaker again. Thank you so much.
What did I do with this? You'll do it. Okay.
Hey. I did pick up work. It says book you all the time and put it on here. I'm very sorry. Okay.